So we are going through inflammation, or we are studying inflammation. And in the previous lectures, we talked about macrophages and other cells. And now we are going to talk about the inflammatory cytokines that cause fever response. Fever is a physiological response of your body when there is some kind of infection, particularly with some bacteria, etc., or other microorganisms. Then the body would generate the fever response, hyperthermia or increase in body temperature. So inflammatory cytokines cause fever and induce the acute phase response by the liver. Okay? What is the acute phase response? You would come to know in a while. Okay? So there are different things that can cause fever response or that can increase the body temperature. Okay? And these things can either come from outside the body, or these things may be produced inside the body. We call them pyrogens. They are pyretic. They increase the body temperature. They cause hyperthermia. So there are some endogenous regions or molecules produced inside your body or inside your cells, and we call them even cytokines. Okay? The cytokines that we have been studying, okay? Particularly this one, interleukin 6. And there are exogenous regions in the exogenous regions, which may be some bacterial infection, for instance, or the products, bacterial cell walls products or membrane products or other organelles when the bacterium is degraded inside the cell. So bacteria can also increase your body temperature, and endogenously these molecules produced inside the cells, which are known as cytokines, also increase the body temperature. And you also have studied about these cytokines, interleukin 1 beta or interleukin 6 or tumor necrosis factor alpha and other cytokines, the pro-inflammatory cytokines secreted by macrophages when they are activated. How the macrophages are activated? When they come in contact with bacteria, they are activated. So they would secrete these different types of cytokines that are going to affect different organs inside your body. For instance, these cytokines produced by an activated macrophage can act upon the liver. Interleukin 6 can directly affect the liver, and the liver would start synthesis of acute phase proteins, Just like the CRP and MBL, in the next slide you would see that what are these proteins. And these proteins also are used just as the complements would be used in your body. Do you remember the complements? The complements are used for labeling of different antigens. So when you label the antigen, this is very easy for the immune cell to phagocytose or engulf that particular antigen. Okay? So these acute phase proteins, which are synthesized by the liver under the influence of this interleukin 6, which is secreted by a macrophage, are used for the same purpose. Okay? They would increase the phagocytosis of the antigens which are there in the body, which is called opsonization. Then these cytokines can also directly affect the bone marrow, and they can mobilize the neutrophils. And if you remember, I had told you that neutrophils stay inside the bone marrow for 5 days. And outside the bone marrow, the neutrophils would just live for a day or two at the maximum. Okay? And the neutrophils, you also know that they are not found in healthy tissues. Okay? Normally they are in the blood. But when there is some bacterial infection, the neutrophils are invited to the site of infection. They would go there. Okay? So this process also is guided or shaped with the help of different cytokines, which are secreted here during this process of inflammation. And this ability of the neutrophils to, when there are more neutrophils, I had told you that neutrophil is relatively, they are smaller cells as compared to macrophages, but they are most numerous cells in the body. So these neutrophils are clearing more of the debris or more of the pathogens from your system as compared to macrophages. The macrophages also are doing other jobs, They are the key players in enhancing this process of inflammation. They are the central cells which are going to involve all other partners in this process of inflammatory response. They are inviting different cells with the help of different cytokines that they are secreting, etc. But the neutrophils, they are specialized cells for killing the pathogens, by different means. They can also enhance this inflammatory process by secretion of different cytokines, but at the same time, they also are going to engulf most of the pathogens, and you know that the neutrophils die after an interaction with the pathogen, These cytokines also are going to act on the hypothalamus. Interleukin 6 can directly, affect your hypothalamus, the thermal center in your brain, and increase the body temperature as a result of that. And not only that, not only by that mechanism, it also can directly affect on the fat and muscle cells, and it can increase the metabolism or the rate of metabolism in these cells, and that would also increase the body temperature needed there. So why this response? This is actually the fever response generated here when the interleukin 6 would affect the hypothalamus or the fat and muscle cells. In both these situations, heat will be produced in greater amount, and that heat response or fever response is going to decrease viral and bacterial replication. Okay? So fever response is normal physiological response of your body, and the body is generating fever response in order to stop the replication of the pathogen, which can be a bacterium, a virus, or other microbe. Okay? That's the process that is going on. And you are taking medication when there is some kind of fever response, you say that I'm sick or I have some problem, okay? In order to subside the fever response. So sometimes this fever response can be very severe because the the body doesn't understand that you can also succumb to this situation if the fever response sustains at a very high level for more, time. Then it can be dangerous as well for the same cell. Okay, that's why you take medication in order to drop this response, which is not needed with that intensity. Okay, and along with that, you use different kind of antibiotics and other drugs in order to help with the cells of your immune system in killing that particular pathogen which is causing this problem. You understand? So the fever response disfavors bacterial and viral replication and protects the cells from being damaged by the effect of certain cytokines like the tumor necrosis factor alpha, which can also be dangerous in some cases if this response is not managed or calculated. The acute phase proteins produced by the liver, you know, there are about 30 different types of proteins that would be produced by the liver, 30 plasma proteins, proteins that are found in the blood. We call them plasma proteins. There are about 30 plasma proteins that would be produced by the liver when it is influenced by this cytokine interleukin 6. Okay? In the same cytokine which is going to cause hyperthermia or increase in the body temperature if it affects the hypothalamus or cells of your muscles or body, fat cells or muscle cells. This cytokine is going to affect the liver to synthesize these 30 plasma proteins. And where is the interleukin 6 coming from? Again, from the inflammatory cells. Okay, the macrophages and cells that are secreting interleukin 6 because there is some infection. Okay, so normally interleukin 6 should not be there in the system when the bacteria is not there. But when the bacteria has, attacked your body, interleukin 6 is secreted by your immune cells, and that is going to do different jobs in different organs. Okay?
Sir,
This is the liver.
interleukin 6 is not produced or it is not formed.
What?
ये एक्टिवेटेड नहीं होता या फिर ये बना नहीं होता?
This is a cytokine. Normally, it should not be released by the cells when there is no situation. It's a pro-inflammatory cytokine. Pro-inflammatory cytokines are released when the inflammatory process is needed to be started inside your bodies. So out of these 30 plasma proteins, the acute phase proteins are all those proteins which would have 25% ups and downs, increase or decrease in their concentration. This is just a rough estimate of proteins. Some of the proteins, the level of some of the liver proteins, it would go up, just like this one, as you see, and some of the proteins level, it would go down. Okay? So acute phase proteins would be proteins produced during this inflammatory response when there is some kind of infection, and their level is, you can say, abnormally elevated during this process of inflammation. Just like the C-reactive protein or the serum amyloid A protein, or you can say fibrinogen here, okay? And you see that these proteins, the level of these proteins is going up, okay, under the influence of interleukin 6. Who is synthesizing these proteins? The liver. Okay? The liver is synthesizing lots of different proteins and immunoglobulins. So, albumin, for instance, normally albumin should be the most abundant plasma protein inside the blood. The most abundant liver protein is albumin. Okay? But when there is infection, then other proteins, the acute phase proteins like the serum amyloid A, the C-reactive proteins, etc., these proteins are the names that you can see here: lipopolysaccharide binding protein, or different complement proteins, or these factors that you see: granulocyte colony stimulating factor, okay, fibrinogen, plasminogen, tissue plasminogen factor, etc. All these are acute phase proteins. Okay, it means the level of all these proteins would go up during infection, but the level of proteins normally synthesized by the liver would go down. Okay, so the liver cell is more committed to make proteins for specific job, and that job is to somehow help with clearance of the microbe, bacteria or viruses or fungi, etc., whatever the infection is. Okay, these proteins are needed for a purpose. So what is that purpose? That purpose is that if you take the example of C-reactive protein, the C-reactive protein is going to bind any bacterium that has entered the body, or a fungi, or a parasite. This C-reactive protein also can bind with phagocytes like macrophages and neutrophils to enhance this process of inflammation, to make them secrete cytokines that can enhance the process of inflammation, or bind to these different pathogens and activate the complement pathway. For instance, the C-reactive protein can, you already have studied it, it can trigger the classical complement pathway, and that pathway also is, finally that pathway would also lead to the clearance of the microbe or killing of the microbe. Okay? There is mannose binding lectin, and you know that mannose binding lectin, this also is used for pathogen recognition. It's going to start the MBL pathway, the lectin pathway of complement activation. Okay? And the lipopolysaccharide binding protein, or LBP, again it's used for pathogen recognition. These complement proteins that you see here and factor B, these also are synthesized in the body for the purpose of eliminating the pathogen. And you know that the complement proteins, they label the cells, okay? The complements can directly kill the microbes, like bacteria, for instance, and they can also indirectly kill the pathogen by just labeling them and enhancing this process of phagocytosis, which is called opsonization. Okay? So all these complement factors or proteins are also synthesized in the liver. Okay? These factors that you see here, like the serum amyloid A proteins, are meant to increase the inflammatory response. Okay? Certain, factors like the fibrinogen or plasminogen or tissue plasminogen factors help with the process of coagulation. Okay? So, what is serum amyloid A protein? This is a small peptide, about 100 amino acids in length, which means it's quite small, and usually inside the blood, this is associated with HDL, okay, high density lipoproteins. And serum amyloid A, the acute phase protein synthesized by the liver, binds with toll-like receptors, and you know that toll-like receptors are the signaling receptors, are the scavenger receptors SRs, and induce the production of cytokines. Okay? Where are these receptors found? For instance, you already know that toll-like receptors are on many different immune cells, are inside many different immune cells, including the macrophages. So if serum amyloid A would bind to a macrophage, okay, or other cells with a toll-like receptor, it's going to activate that particular immune cells. The macrophage would be activated. It would start secretion of different cytokines, just like interleukin 1 beta or interleukin 1 alpha and other cytokines would be produced by the macrophages. Okay? So this is about the acute phase proteins, that these proteins are synthesized by the liver when there is some infection, and how the liver would come to know about that infection somewhere in the tissues. It would come to know with the help of cytokines that are produced by immune cells in the tissues, and then these cytokines also have systemic effect. Systemic effect means that these cytokines would just reach there to different organs with the help of blood circulation, and then they would just do their job.